Hello, my name is Jessica Palmer, and I am the Associate Director of Toxicology at Stemina Biomarker Discovery. Thank you for stopping by our poster, A Human Pluripotent Stem Cell-Based Assay, devTOX quickPredict, accurately and reproducibly predicts the developmental toxicity potential across a diverse set of chemicals. In this poster, we present data from over 100 compounds that have been tested in the devTOX quickPredict assay. devTOX quickPredict is a human pluripotent stem cell-based assay that predicts the developmental toxicity of a compound based on changes in ornithine and cysteine metabolism. In order to understand the applicability domain of the assay, we have separated the results into different pharmacological categories and assessed their performance. We have also evaluated the reproducibility of our predictive model using independent replicates of carbamazepine, methotrexate, and thalidomide from the last 5 years. Moving on to the methods, this assay is conducted with human pluripotent stem cells that are commercially available from ATCC. The cells are maintained in an undifferentiated state and plated into 96-well plates where each chemical is assessed at 8 concentrations. In addition, each plate includes a positive and negative control that are used to ensure the cells are metabolizing ornithine and cysteine at a level that is consistent with how the assay is developed. The cells are exposed to compound for 48 hours, and then the spent media is collected and analyzed using a targeted LC-MS-based method. We will also do a cell viability assay on the cells to compare with the changes in ornithine and cysteine metabolism. This data is then fit with a nonlinear dose-response model, and the concentration where the metabolism in the cells changes is predicted to understand the developmentally toxic and non-developmentally toxic exposures for a given compound. To evaluate the accuracy of our assay, we've used 2 prediction models. In the first model, we will compare the developmental toxicity potential concentration, or the point where the OC ratio crosses this developmental toxicity threshold, to an in vivo exposure. These exposures are based on human data or in vivo data where a compound is known to be developmentally toxic or known to be not developmentally toxic. Since this type of data is not available for all compounds, we have also set a concentration threshold using our training set to separate non-developmentally toxic from developmentally toxic compounds. When we set this threshold, the optimal concentration was found to be 65 micromolar. So if a compound has a DTP above 65 micromolar, it's predicted to be non-developmentally toxic in the assay. If the DTP is less than 65 micromolar, it's predicted to be developmentally toxic. Looking across this broad set of chemicals that have been tested that range from pharmaceuticals to naturally occurring environmental chemicals, the accuracy of the assay is 87%, with a sensitivity of 88%, a specificity of 86%, and a positive and negative predictive value of 88 and 86% respectively. If you look at the notes section here under the table, it identifies which prediction model was used for each of the compounds, if you're interested. We've also taken the data and separated it into the different use classes to understand or demonstrate how the assay performs across this diverse set of chemicals. To understand the applicability domain, we've separated the pharmaceuticals into different pharmacological classes, such as channel modulators, DNA modifiers, enzyme modulators, kinase modulators, and so on. We've then assessed the accuracy, sensitivity, and specificity for each of these different classifications. As you can see, in general, the assay performs very well across the board. There are a few areas where the sensitivity is lower than the other classes, and I'll get into that a little later in the poster. When we look at the reproducibility of the assay over time, you can see that there is some variation in the DTP potential, or the DTP concentrations for these 3 compounds that we use, but all of the DTP concentrations fall within 2 standard deviations of the mean. And all of this variation is really what it would be expected based on biological differences over time. At the top of the third panel, we show a number of different examples of dose response curves from the different types of pharmaceuticals that have been tested in the assay. As you can see, there are a number of different compounds and different response types that can be observed. All of these compounds were compared to either their in vivo rodent Cmax value, where a developmentally toxic effect was observed, or compared to the human Cmax value, where developmental toxicity was observed. Under receptor modulators, I show 2 examples, one of a compound that is predicted to be developmentally toxic at biologically relevant concentrations in vivo, and one where the prediction is incorrect. What is interesting is fingolimid phosphate is a sphingosine 1-phosphate receptor inhibitor. Looking at the gene expression data for human iPS cells, it indicates that the undifferentiated cells do express one of these receptors. So it's possible that the effects that we're observing in the assay are linked back to changes or inhibition of the target receptor. In contrast, bosentan isn't causing an effect in the assay until just above over 10 times higher than the rodent Cmax. The gene expression data for the bosentan target, which is an endothelin receptor, indicates that the cells do not express or have very low expression of these receptors, which could be why the cells are not responding at biologically or pharmacologically relevant concentrations. In conclusion, in this poster, we've tried to demonstrate that the devTOX quickPredict assay is a highly accurate method for predicting the developmental toxicity for a diverse set of chemicals. The assay is able to identify developmental toxicity for a wide range of mechanisms, but it may not be applicable in all cases, especially where the target of interest is not expressed in the pluripotent stem cells. We have also demonstrated over time that the assay is reproducible across multiple reagent lots and technicians. And the next things that we're working on are to finish testing the ICH reference compound list and incorporating in vitro to in vivo extrapolation with the data that's presented here. We hope to have a publication out later this year that includes the data from all of these chemicals and the additional compounds from the ICH list for everyone to share with everyone. Please let me know if you have any questions. I will be available at Stemina's virtual booth for questions on this poster or on any of our assays in general. And I hope everyone's having a great SOT.